Hi everybody, now we're going to talk about lipids and membranes. Just as a disclaimer, I'm much better at this lecture when I can use my hands. And I even had like a live demonstration in class last time where people lined up and threw Nerf balls at each other. It was actually pretty cool, so I don't know how well this will translate on screen, but we'll give it a go. Okay, so we're going to talk about lipids, structure, how they're made, and most of the time though we're going to spend talking about membranes and how chemical compounds get across membranes. Okay, so lipid is a catch-all term, basically for organic compounds that are very largely nonpolar and are principally hydrophobic. So most lipids are hydrocarbons, meaning that they're essentially made of carbon, hydrogen, and not a whole lot else. There are many lipids in the world. We're going to stick to talking about biologically relevant lipids. Most biologically relevant lipids contain something called a fatty acid, which is a hydrocarbon chain coupled to a carboxyl group. Hopefully you remember the term carboxyl group because we discussed it a few videos ago. So, sorry about that. So what we see here are diagrams of two different compounds. This is called isoprene. This is a fatty acid, which we mentioned a second ago. So the point here is that these hydrocarbon chains can be very short or they can be very long. And when they're long and coupled again to a carboxyl group on the end, these are fatty acids, and these are the ones that are biologically relevant. So lipids aren't defined in the same way that DNA or RNA or proteins or carbohydrates are. They're not defined by their structure or by how they're made. They're defined by their solubility, namely that they're not soluble in water and that they're therefore hydrophobic, meaning water-fearing. By the way, anything, just as a reminder, that is soluble in water is called hydrophilic. But I put this, these two images up here to show what happens on a molecular level when a lipid is dropped into a solution of water. You get this complete ring and lack of interaction between these molecules and these ones. And the way that we see that on a grand scale would be something like if you see a puddle with oil in it, or if you have, a bottle of salad dressing that's a vinaigrette that needs to be shaken up before you can actually use it. You can see that the oil components versus the aqueous components do not mix together. Lipids are, on the other hand, soluble in organic solvents such as ether. So if you have not yet taken organic chemistry, you'll find that in organic chemistry lab, when you're dealing with hydrocarbon compounds, you nearly always have to use ether in place of water. Okay, so to keep this biologically focused, we're going to talk about three different types of lipids that are found routinely in cells: fats, steroids, and phospholipids. So fats are basically just three fatty acid chains linked to a molecule called glycerol. I'll show you how that works in a second. We're going to talk about steroids, which are, have a big ring structure and a tail on them, and then finally phospholipids, and principally we're going to talk more about phospholipids than any of these other kinds. Okay, so to get to fats first. When you're synthesizing a fat, you have a fatty acid chain here, you have a molecule called glycerol here, you have a free hydroxyl group here, and a hydroxyl group here. So guess what happens? Once again, when these are being formed, we get a condensation reaction between this hydroxyl group and this hydrogen, and you get a water molecule produced, and you have this fatty acid being linked to this glycerol. To make a complete fat molecule, you have three fatty acids linked to these three original hydroxyl groups on the glycerol molecule. And so the fancy name for a fat molecule is a triacylglycerol. There you have it. Impress your friends. The next one that we're going to talk extremely briefly about our steroids. So we have basically these 4 hydrocarbon rings and a tail of something called isoprene, which we saw on the first slide. The point, and the reason why I wanted to bring up steroids are not only because they're tend to be important components of cell membranes that help hold them together. Cholesterol is a great example of a steroid membrane, a steroid molecule that's used to hold a membrane together, but because of this. Steroids tend to have an R group attached to these hydrocarbon rings that is usually polar. The steroids themselves and the isoprene chain are nonpolar. So this is the first molecule we're seeing that's got a head and a tail to it. It's got a very, very strong polar end and a very strong nonpolar end. So phospholipids are very chemically diverse. They consist of a fat molecule, a phosphate group, and just about anything else attached to it. They also have these big long fatty acid chains at the as their tails, with one exception. Fatty acid chains are the tails of phospholipids for bacterial cells and eukaryotic cells. For archaeal cells, their tails are not fatty acids but are isoprenes instead. Just like steroids, phospholipids have a polar head portion and a nonpolar tail portion. This makes them extremely good at spontaneously forming membranes. And in fact, cell membranes, even though we pointed out in the previous slides, they're helped to be held together by steroid molecules. Their primary component are phospholipids. Okay. So membranes, cell membranes, are once again basically composed of steroids and phospholipids, and these are amphipathic. That's the fancy way of saying they have a head and a tail, or they have a hydrophobic end and a hydrophilic end. What is going to happen is that when you put a whole bunch of steroids and phospholipids together, all of the heads are going to interact with each other and all of the tails are going to interact with each other. The tail of one is not going to interact with the head of another one. In addition to that, the other point we can take away from this is that the heads are going to interact with water or other polar compounds and the tails are not. The tails are going to absolutely avoid water. So the cool thing about membrane lipids and membranes in general is that they form spontaneously, and we'll talk about that more on the next slide, but you wind up with them all being perfectly oriented in the same direction, in that all of these heads are only going to interact with each other, so you're not going to have random upside down phospholipids. All of their heads are going to be pointing in the same direction. All of their tails are going to be pointing in the same direction. What this is a picture of here is what's called a phospholipid bilayer, meaning there's a layer up here and there's a layer down here. All cells are encased by phospholipid bilayers. And the other point you can take away from this is that you have hydrophilic condition on the outside, hydrophilic condition on the inside, and about 9 miles of hydrophobicity in between. Okay, so phospholipid bilayers as they apply to cells. You get these nice sheets of phospholipids that line up again in the proper orientation. You have one sheet lining up here pointing down, another sheet lining here pointing up, and the way that you again don't get this one kind of flipped on its head, so to speak, and have these tails lining up with these heads is that again these heads are not going to interact with these tails. These tails are going to interact with each other and these heads are going to interact with each other, and that's it. Once again, I mentioned this a second ago, but these happen spontaneously. They form all by themselves and you don't need any energy to build this entire membrane. The other thing that's kind of cool about membranes is that they're constantly in motion. So this head right here isn't covalently or even ionically bonded to this one. It's just attracted to it. So these guys are not chemically bonded together at all. And that means that membranes are extremely fluid and floaty, and things can move in between them because there aren't permanent bonds between them. As we said a second ago, when we were pointing at what could be a cell membrane, we said there's a hydrophobic conditions up here, I'm sorry about that, hydrophilic conditions up here, hydrophilic conditions up here, down here, so outside the cell, inside the cell, and a whole bunch of hydrophobicity in between. It's almost a bit unsettling, isn't it, in that animals don't have cell walls, we're animals, and so the only thing separating the inside of our cells from the outside is this bilayer membrane that's not even held together by any chemical bonds other than attraction. It's almost a little unsettling, but it's kind of cool. Okay, so again, if we're looking at a phospholipid bilayer as a cell membrane, typically cells are in aqueous solutions, so basically salty water. Cytosol or cytoplasm, when the inside of the cells are also basically just salty water. So the heads are going to be oriented towards these outsides and the tails are going to be oriented inward. Okay, so for the rest of this time we're going to talk about permeability of lipid bilayers. And what I mean by permeability is the tendency of any given substance to be able to move across a membrane. Phospholipid bilayers are very, very effective cell barriers because they have very selective permeability. So not just anything can move across a membrane. The first thing to talk about then is who gets into a cell? And the corollary to that is who gets out of a cell. Basically, small things, non-polar things, that's pretty easy. Charged things, very large substances, they very rarely can move across a membrane all by themselves. They need some kind of outside help. Okay, so before we talk about how that actually works mechanistically, I have one more point about the types of fatty acid chains that are in lipid membranes, that are in membranes, and what that has to do with their permeability. So if you have a hydrocarbon chain and it's got no double bonds in it, it's all carbons single bonded to themselves and only to other hydrogens, you've got the absolute maximum possible number of hydrogens in that chain. In that case, that's called a saturated fatty acid. If you do have double bonds in your carbon, you don't have the maximum possible number of hydrogens, and you are called unsaturated. When you are an unsaturated fatty acid, what that does kind of geometrically is it puts a kink into the fatty acid chain. So what that looks like from a phospholipid standpoint is you have this polar head here. You have usually one saturated fatty acid chain that's nice and straight, and you have an unsaturated fatty acid chain that's kinked out. So if this would be the double bond here, this would be the actual molecular diagram, this would be the phospholipid diagram. You can see everybody's got this little kink in it if this is an unsaturated chain. Sidebar, this picture totally reminds me of the green M&M in commercials, I'm just saying. The reason why one would care about saturated versus unsaturated fatty acids has to do with the geometry of the membrane itself. If you have a bunch of phospholipids that are exclusively made of, that exclusively have saturated fatty acid chains, those phospholipids are going to be able to pack very, very tightly together. If you have some unsaturated fatty acids in there, that kink in the leg, if you like, is going to keep those phospholipids from packing as close together as if both were saturated. And so the outcome of that is that the membrane is much more fluid, it's much more porous, and it's much more permeable. In case you couldn't predict this, this was the point at which I had people kicking their legs out standing in a line and other people chucking Nerf balls at them to show that they could get past them when they were standing further apart, and they couldn't get Nerf balls past them when they were standing very close together. Anyway, that was a fun day, but I digress. So we can actually see, this is much more obvious when you look at this picture, we can actually see in sort of everyday objects how this comes into play. So if you have exclusive saturated fatty acids, you're not going to be terribly permeable because these are squished very close together. If you have unsaturated, they're not squished quite as closely together and you're much more permeable. Our everyday objects that are great examples of this are things like butter and beeswax and plant oils. As we all probably know from the news and reading scary nutrition stories, butter has a lot of saturated lipids in it, and as an effect and an impact of that, you can see it's solid at room temperature. You can't have anything pass through this. Same with beeswax. Beeswax is full of saturated lipids with very, very long hydrocarbon chains, none of which are kinked, and this is completely solid. You can't poke anything through that. On the other hand, if you look at safflower oil that has unsaturated lipids in it, you can turn this bottle upside down, it'll swish around, it's very fluid. You can drop objects into it and they'll move through the fluid matrix. It's very, very obvious when you think of these three things why this would result in the membrane permeability that it actually does. Okay, so movement across lipid bilayers. How does this actually work? The first thing that happens, and the easiest way that this happens, is if we're talking about these small molecules in a solution. These are called solutes. They can move across a membrane by simple diffusion. So this is just random movement of molecules due to kinetic energy. That's it. If they're chemically able, they will float across this membrane all by themselves. And they'll do so in response to something called a concentration gradient. So if there's a whole bunch of a certain type of molecule out here and not very many in here, they will move from the higher area to the lower area until they're equalized out. So the point of that again is that solutes move from regions of high concentration to lower concentration across the membrane. If they're able to do that all by themselves, that's great. Okay, so just to put this in, to give this another example, if we have two different types of solutes in a beaker that's separated by a synthetic membrane that's permeable, if we have, say, purple triangles and white balls, what is going to happen is that these are going to start diffusing, both of these, across the membrane, and ultimately what you'll have is on either side of this a solution that's half purple triangle, and the other solution is also half purple triangle, and then both are also half white ball. So when you have this situation where you've got the same concentration of solute on either side, that's called equilibrium. And the other point of this figure being set up the way it is that this is one single membrane, and it at the same time is allowing for diffusion of two different types of molecules across it. So the purple triangles diffusing across have nothing to do with the white balls diffusing across. They're completely independent events. Okay, so it's important to point out at this point that not everything can move across a membrane by simple diffusion, and when it's important that equilibrium be established, water can move across a membrane. So osmosis is this movement of water across a permeable membrane. Osmosis is a type of diffusion. That's a very important thing to know. And in osmosis, the water is moving, not the solutes. So if you have different concentrations over here, One way that they can be equalized is if, again, like we saw in the previous slide, if these green balls move over here until we have the same concentration on either side, but if the green balls aren't able to cross this membrane, the water molecules can. So ultimately what happens is that some of this water will cross this membrane until we have the exact same green ball per unit of water concentration on either side. You're going to wind up with a different volume on either side, mind you, but this concentration is equalized. So osmosis is particularly relevant to cells because cells tend to sit in aqueous solutions. Osmosis is not always real good for cells to undergo, however. If cells are in something called a hypertonic solution, this basically means they have more of any given solute on the outside than they do on the inside. And what happens is that the water molecules inside of a cell cross this membrane in an attempt to equalize the concentration, and this cell ends up dehydrated. It ends up shriveling up. On the opposite side of the coin, you can have a hypotonic solution, which means you have a lot of solute on the inside of the cell, not very much on the outside. So what happens here, again, is that this water now crosses this membrane, and the cells start to swell up, and eventually, if this goes on long enough, they can burst. Obviously, that's also bad. Finally, what you ideally want is your cells to be in something called an isotonic solution, where you have equal concentrations of solute on either side of the cell membrane, and then the cell is happy. It doesn't shrink and it doesn't burst. It's just there. Okay. So that bit of discussion involved simple diffusion, things that are able to cross a membrane by themselves. However, not everything is, and in fact most things aren't. So when we're in this kind of a situation, we're going to have to rely on some proteins that are embedded in the membrane. So when we talk about cell membranes, typically the phrase fluid mosaic model comes up. Hopefully everybody can see where fluid comes from. The idea that, again, these phospholipids are not chemically bonded to one another and they can float around and move. The mosaic aspect of this has to do with proteins that are embedding in the membrane. These guys, it's not like this protein sits up here and is permanently in this location. It can actually float through this membrane and move around in and of itself. So a cell membrane is a very dynamic thing. It changes the way it looks and the way it's shaped. At this point I want to describe that there are two different types of membrane proteins. There are integral and there are peripheral. Integral membrane proteins are in and of themselves amphipathic, meaning they have they have hydrophilic pieces to them that are going to interact with the outside of the cell and with the heads of the phospholipids, or the inside of the cell for that matter. And they also have hydrophobic regions. These are the ones that are interacting with the tails of these phospholipids. Because these proteins span across the entire membrane, they're frequently, more often than they're called integral membrane proteins, they're called transmembrane proteins. So if you hear that term, that's what it means. So in addition to integral or transmembrane proteins, what you can have are peripheral membrane proteins. These guys do not span the membrane. What they do is that they're found on one side or the other, and they're attached to integral membrane proteins. So this guy, this peripheral protein right here that's facing the inside of the cell, if this protein were not sitting here, this peripheral membrane protein would not be interacting with the membrane. Neither would this one that's facing the outside. Okay, so membrane proteins, this is kind of a misleading title to the slides. There are many other things that membrane proteins do, but in the context of this conversation, membrane proteins control transport. Okay, so cells need stuff, and stuff needs to get in. And if the stuff that needs to get in can't get in by simple diffusion, you're going to need some kind of help. I don't have this on here, but you might also be able to guess that stuff also needs to get out of a cell, and if it can't diffuse out, it's going to need transport proteins for help. So transport proteins come in three different types that I want people to be familiar with. One are called channels, another are carrier proteins. These are also called transporters, and then the last one are pumps. Channel proteins participate in something called facilitated diffusion. This is where you have small charged stuff that needs to get in or out of a cell. And channel proteins tend to be stuff specific. So what I mean by that is you can have a channel protein that's specifically there to allow water molecules to pass through, and it won't allow other molecules to pass through. The way that these work so well is that these are integral membrane proteins, and they almost have two layers. The first layer that interacts with the actual fatty acid tails of the phospholipids is completely hydrophobic. So, hydrophobic exterior. The hydrophilic part, or the inside part that interacts with the stuff that is meant to channel in and out, is hydrophilic. So these things that wouldn't be crossing a membrane in the absence of this channel are able to, because the channel is hydrophilic on the inside. An important point about channel proteins is that this is not an energy dependent process. This is called passive transport. It's not simple diffusion because they're not going through the membrane by themselves, but this is called, this is considered passive because it's happening in the direction of an electrochemical gradient. It's going to go from regions of more to regions of less in the appropriate direction, but again with no energy input from the cell. Since we have no energy input, I feel it's important to point out that this would not happen spontaneously, and the question to think about is, why would this not happen spontaneously? Pause, think, see what you come up with. But the answer is that everything passing through the channel is not going to chemically be able to interact with this 9 miles of hydrophobicity here. And so it needs this hydrophilic interior to be able to move across by itself. Okay, so that's why this wouldn't happen spontaneously. On the in the same vein, you have carrier proteins. These are also an example of proteins that mediate passive transport or facilitated diffusion. So these guys transport solutes from regions of more to regions of less. Again, they follow the electrochemical gradient. Very much the same as channels. They don't require any energy, but this transport would not happen spontaneously because, again, these solutes would not be able to interact with this hydrophobic region. They'd get stuck. The way that this works as opposed to a channel protein, so this is where hand gestures come in extremely handy, is that these channels will sit with one direction open. When some solutes feed into them, they change orientation. This and this side are going to flip together, and then this solute's going to spin out on the other side. As opposed to channels and carriers, we have pumps that sit in membranes. These are different. These employ what's called active transport. Active transport is when you're moving molecules against a chemical gradient. So this is where you're going to require some energy to do that because that's not going to happen spontaneously. The way that they work, there's a lot of details here, but they create an electrochemical gradient across the membrane falsely, and then they allow solutes to be tricked into moving into from lower areas back into higher areas. A cell intentionally will use a membrane pump to mediate its own environment in a way that's favorable to it. That sounds very convoluted, but it's an extremely it's an answer based extremely in physiology that I'm personally not qualified to answer all that well. Anyway, the point being, this diagram represents what you would expect would happen if there were a channel or a carrier protein sitting here. We have a lot of sodium on the outside of this membrane and only a teeny bit inside. We would expect that sodium here would start flowing in. That's the natural direction it would go. That's the way the electrochemical gradient points. What actually happens if we had a membrane pump here is that it will take this region of, sorry, this is a bit flipped on its head, but you would take this region where you have a lot of sodium, and you're going to take this region where you have a little bit of sodium, and you're still going to have this remaining molecules pumped out to this side. Same thing with potassium in this case. You have a little bit here, you have a lot here, and you want to swap these two salts one for the other. This is an example called the sodium-potassium pump, and you're going to pump this potassium inside here, even though the electrochemical gradient dictates it should flow that way and that way respectively. If the cell has some physiological reason for wanting to get the sodium out and the potassium in, it's got an energy requiring mechanism to do that. Okay, so in summary, we have three different kinds of membrane transport. We have diffusion, facilitated diffusion, and active transport. Diffusion, once again, is this passage of compounds across a membrane in the direction of an electrochemical gradient all by themselves. Whatever's moving in and out is able to fight its way through the membrane on its own. Facilitated diffusion, again, no energy required. You're going again in the direction of an electrochemical gradient, but you require either a channel or a carrier protein to do it, because whatever's flowing in and out does not interact with the membrane very well. And then finally, active transport is an instance of going against the electrochemical gradient, and it does require energy input from the cell. That's all I've got about lipids. Next we're going to start talking about actual bacterial cells.